Thanks so much. Thank you for inviting me to share this paper today. So I'm going to be talking about application of pooled CRISPR screen with imaging of our work that was recently published. And this work has been done co-led by another student in the Yale lab, Anthony Vu. And I think it's a fun example of a collaboration between someone who's really interested in the cell biology and biological phenotype of stress granules, which is the expertise that Anthony brought to this project, and myself, where I had this idea to kind of adapt pooled CRISPR screening to image-based phenotyping, which is really, as we all know, a very, very important aspect of cell biology. So this project was a joint effort between the two of us, and as Matthew mentioned, I did my PhD in the Yale lab and just recently have accepted a postdoc position at the Dana-Farber, and will be starting as a postdoc there in a couple months. So jumping right into things, I think that we can all appreciate how important image-based phenotypes are to tell us what is going on in cellular behavior. So myself, as well as many others, have looked at sequencing data, and we can very easily get sequencing data and use that to learn information about gene expression or transcription states of the cell, or what's going on in the nuclear level. And while it's really great because we can access this information easily and in high throughput, with sequencing data, we're often trying to infer what that means about cellular behavior, and we're making guesses that we kind of have to eventually go validate in the lab. But I think imaging is really, really cool because rather than trying to infer something, you can simply look at it. So here is an image with a bunch of different examples of cellular behaviors that you can reveal through protein or RNA staining, or even whole organelle organization, that can tell you something about what's going on in the cells and what genes are important to control these various behaviors that we can see. And so what I was really interested in doing is not just being able to look at where the proteins or RNAs may be localized in the cell, but to do a functional genomics platform to be able to figure out what are the genetic regulators of these various phenotypes that we're seeing. And this project was really born because our PI Gene, he wanted to do a screen looking at what the genetic regulators are of stress granule formation and stress granule assembly. However, these functional genomic screening platforms are often really laborious and require complicated setups, the use of robotics, so that when you are performing manipulations in thousands, potentially thousands of wells with replicates, to make sure that these very small volumes are accurately pipetted, you don't have batch effects, and it's often a very complicated process. These types of screens are most successful in large core institutes or facilities that have robotics platforms to be able to do this. And so we were trying to think creatively about different ways that we can approach functional genomics with imaging-based phenotypes that can be applied really in any laboratory setting using kind of common materials that are available to us. And we thought that this gave us a really good opportunity to kind of expand the world of CRISPR screening, because as we all know, those are really useful methods to be able to perform genetic knockouts in a simple and easy to use fashion. So these genome scale CRISPR-Cas9 libraries, which have since been adapted to much smaller scale libraries, have really revolutionized our ability to perform gene knockout in cells. So having oligo arrays that are easy and cheap to synthesize, which are now often available in a pre-pooled format in their lentiviral vector. You can then take these array of oligos and clone them into a lentiviral library where each particle of lentivirus contains the genetic information to perform a unique knockout. And so if you do this and you infect cells at a low multiplicity of infection, you hope that, and in most cases it's true, where you only get a single lentiviral particle per cell. And this allows each cell that you infect to have a unique genetic knockout. But the problem with this, or maybe the beauty of it for how it started, is that now you have this pool of cells remaining, each of which are different and unique. And so how can you analyze this pool of cells for something that is meaningful and useful to tell you about behavior? So the first thing that was used is a simple growth phenotype where you can grow these cells in bulk over the course of maybe 2 weeks, and you can see which guide RNAs deplete from the pool, and those would represent guide RNAs targeting genes that are essential for cell survival. So these are survival-based screens, which you can do under particular genetic conditions for synthetic lethality, et cetera. We can also use fluorescence reporters in the high throughput assay of FACS sorting to be able to do this in kind of a more interesting setting, maybe a little bit more specific to a phenotype that you're interested in. This can be applied and has been applied in vivo as well, where you have cells that are genetically engineered with a specific fluorescent reporter. And then upon treating them with this lenti-CRISPR library, you can then allow them to grow in their natural environment and select for ones that come out at the end, which have either high expression of a specific gene or low expression of a specific gene. However, this does require the ability to have your readout be FACS-based, where total cell fluorescence is what you're interested in. And as we all know with imaging, it's often not about total cell fluorescence, but rather more specifically where in the cell something may be localized. And so the other options that have come out recently using these lenti-CRISPR methods are to combine with various transcriptomic approaches. So single cell transcriptomics allows you to more specifically combine the application of what a particular guide RNA is along with the single cell transcriptional profile for a given cell. And then you can use various computational approaches and again, some guessing and inferences to look at gene expression from what you can capture at the single cell level and try to understand kind of more globally what pathways are upregulated or downregulated, et cetera, or how different genetic knockouts may or may not cluster together. And again, these are all sequencing-based, or this particular class would be sequencing-based methods and readouts where you can sequence both the single guide RNA as well as the transcriptome at the same time. And this can have obviously very useful applications. However, when we started this work, there was no application of using pooled CRISPR screens to perform image-based phenotyping that we were interested in doing. So in the context of stress granules, which I'll explain in detail in a little bit later, we care a lot about the localization of a particular protein, not the whole cell expression of that protein. So this is something that we can't get with either a total fluorescence readout in FACS or even a sequencing-based readout of gene expression, because it's not about how much total gene is expressed, but rather specifically where that gene is expressed. And so since we've been doing this, some others have as well, and some really cool technologies have come out to use pooled imaging approaches with in situ sequencing. So the first, or this recently published paper performed a screen for nuclear versus cytoplasmic localization using image-based phenotyping, where the guide RNA was read out in a given cell using in situ sequencing. So using probes that are specifically labeled with a different fluorophore allows you to get the actual sequence of the given guide RNA infected in each cell, as well as whatever image-based readout you're looking in. So in their case, they were staining for a transcription factor and looking at nuclear versus cytoplasmic localization. And this work is done in very high throughput. So you can take a plate of cells that are fixed on a plate, and you can image all of them as well as sequence all of them very easily using this approach. However, you have to do in situ sequencing, which it can have a bit of a complicated imaging setup and sequencing setup. The sequencing itself is done actually on the plate, where you wash materials for each round of nucleotide identification across the plate, and then reperform imaging after every round. So this can be quite involved, but it has the benefit of being very, very high throughput. The other application that came out recently is looking at a specific localization of an mRNA using the MERFISH approach of combinatorial flow of fluorescently labeled probes. And this was really cool because in order to get a really nice localization of a given mRNA in the nucleus, or in this case, I guess a long non-coding RNA in the nucleus, it can be done at very high resolution with these MERFISH techniques. However, the imaging setup and sequencing setup can be quite complicated, as the microscopy needs to do this type of MERFISH approach with many different fluorophores at once. It's still a little bit complicated and is not easily adaptable to many labs. And then here you have an issue of throughput where you go much higher resolution. So I think imaging for merfish is typically done at 40x. And then you lose the number of cells that you can capture in a given window. So then your screening capabilities in terms of the number of cells or the number of guide RNAs that you can then screen are limited. And so the particular phenotype that we are interested in analyzing, which we didn't think was applicable, well, I guess that we wanted to adapt a new platform for, is this problem of protein aggregation that occurs in neurodegenerative disease. So I'm just going to give a brief intro into what the phenotype is that we're interested in and how our screening results can teach us something about neurodegenerative disease. So there are many examples of RNA binding proteins which either contain point mutations or mislocalization patterns that we believe is responsible for contributing to the death of motor neurons in disease. So as shown in this cartoon here, this orange cell represents what would be a normal localization pattern of an RNA binding protein, where it's either diffuse in the cytoplasm or even localized in the nucleus under normal cellular conditions. However, it's well known that when you look at patient samples post-mortem, you see that there's these hallmark patterns of aggregations of RNA binding proteins, as well as their target RNAs and a bunch of other proteins that occur in the cytoplasm. And this is a phenotype that is not normally seen in a non-disease condition. And so you can stain for these things post-mortem, where you can take tissue samples and you can see that particular RNA binding proteins have lost their nuclear localization and instead have translocated into the cytoplasm or form these massive aggregates that are potentially causative or correlative with disease of these particular neurons. It's not entirely clear exactly if they're causative or not, given that we don't really see these things until after the patient has died, and this is kind of a fixed end time point. And so what we can do in the lab is try to model how these aggregates form and use that information to try to learn more about whether or not these aggregates are actually causative of death, or if you have any potential therapeutic angles to be able to help prevent the death of motor neurons. And so the way that we and others have been modeling this process is by looking at stress granules, which are these things that normally occur in cells where you have an RNA with its translational machinery is doing its thing in the cytoplasm. And under particular cellular environments, that you get promotion of these things, which we call stress granules, where the translational machinery, as well as its target mRNAs, assembled together in a membraneless format using protein-protein interactions to form these granules and these dense aggregates where translation is halted in the cells. And so this would be a particular stressful environment where cells are maybe trying to conserve energy. And so they form these really nice membraneless separated aggregates. So our lab has kind of developed this model where we believe under normal conditions, this process, we know it occurs and it's transient. So once the stressful condition goes away, we can show that these granules will dissipate and translation resumes and things go back to normal. However, in the context of particular genetic conditions, or as we have shown in previous work, ALS-associated point mutations, you get over-aggregation of these granules where they form normally at the same rate that you would normally expect. However, when you remove the stress and you remove the stressful environment, when you would expect them to go back into their free state and allow translation to occur, they don't. They maintain these aggregates. And this is a problem because if these aggregates form long-term, cells can't translate their mRNAs and make proteins, yada, yada, and they'll ultimately end up dying. So we can see this in a dish in the lab by staining for a core stress granule protein called G3BP1. And this protein normally has a diffuse localization pattern in the cytoplasm, as shown by the green staining here in this image. And when we add an artificial stressor, there's many. The one that we choose and is the most robust is sodium arsenite. And when we treat cells with sodium arsenite, you see these discrete puncta that form in the cytoplasm. And so this is the phenotype that we're interested in screening against. It's well known through mass spec studies that these aggregates contain a lot of RNA binding proteins as well as their target RNAs. And at least in the lab, they always stain positive for this canonical marker, G3BP1. So what we wanted to figure out with this work is not just which proteins are in here or how they form, but we wanted to understand if we could identify any new RNA binding proteins that regulate the rate of formation and disassembly of these granules. And ultimately, can these RNA binding proteins be manipulated to prevent overaggregation in disease? So we're looking for genetic regulators that would prevent the formation of these stress granules in cells. And now our work, as well as many others, have first been studying these aggregates with mass spec-based approaches where you can use proximity labeling or purification methods. And these have been really useful to be able to understand what the proteome is, what the components are of these granules. And that's actually how this work in this paper kind of started, because our lab had done a proximity labeling approach to identify all of the proteins that are enriched in stress granules. And then this is where Jean wanted us to do a screen in kind of an array-based format to take each of these components, systematically knock them out one at a time, and see which of them actually had an effect on stress granule formation. But we thought that we could do a little bit better by expanding this to all RNA binding proteins, because it's possible that there are genetic regulators that are not specifically localized within the granules themselves, but rather upstream or downstream factors, which could be playing a role in their assembly. So by doing the functional genomics screen, you can look beyond what is simply enriched in the granules themselves for other cellular regulators of what's happening. So that's what we were doing in our work here. So here's kind of the general question that we started with. We generated a lenti-CRISPR library that targets all RNA binding proteins. So our lab has a lot of interest in studying RNA binding proteins, and we've used this library for various applications in the lab now. We have an annotation of over 1,000 RNA binding proteins, and this library has 10 guides targeting each RNA binding protein. So it has a lot of coverage per gene, which is really nice. We also have a bunch of positive and negative controls depending on the various applications that you're doing, which you can use to make sure that your screen is working. So this is a targeted library, meaning it's much smaller than the genome scale, and it contains over 12,000 single guide RNAs. And so using the standard methods that others have developed, we take this library, we clone it into lentivirus, and infect cells with this library. I guess I should note that the backbone that we use is standard. It's just the lenti-CRISPR v2. And as opposed to the imaging, in situ imaging-based approaches where they require that the backbone is modified to be able to sequence the guide RNA in situ, ours is using the standard guide RNA backbone.
Yeah, Emily, could you quickly say a word about the genes that are targeted by the positive controls?
The positive controls are known essential genes that have been annotated to cause cell death. So positive controls, when targeted successfully, will cause the cells to die.
Thank you.
We also have some control guides that target fluorescence genes in there. So if you have like a GFP-based screen or something like that, we have guide RNAs that are targeting GFP, which would allow you to, again, have a control for what would be a negative selection in there. And so then again, with this library, now we have a pool of cells in a dish, all of which are different. And the question that we want to be able to do is take this pool of cells, treat them with our stress, which is sodium arsenite, and look to find the rare components of these, or the rare cells in this population that do not form granules, as shown by these yellow cells here. And so the way that we chose to go about doing this was to adapt this platform that has been developed by Nancy Alberton's lab and since commercialized by this company, Cell Microsystem, to use microRaft arrays. And we thought that these were really cool because they're quite easy to use. It's a simple cell culture dish with a media reservoir in the middle. However, when you zoom in, what you can see is that this reservoir contains a PDMS array with a bunch of different individual, you can think of as cell culture dishes, physically isolated from one another in space. However, they share a common media reservoir, so they're really easy to work with. So there's fluid exchange among all of the wells. So if you want to change media on these arrays, you simply suck up the media that's there, and in one pipetting swoop, you add new media, and it will immediately cover all the arrays, or all of the wells. So this is really nice because then you don't have batch effect issues for your individual pipetting. The arrays that we chose to use contain 40,000 microwells in this size. So it's about a 1 square inch array containing 40,000 microwells, each of which are 100 microns in diameter. So these do come in different sizes, and the company and Nancy's lab can kind of customize them. We chose this size because it was good enough for us to be able to grow a robust colony of 293 T cells to be able to screen for. And we wanted to increase throughput. So we wanted to be able to analyze as many different wells as we could. And so the way that you plate cells in here is using math of limiting dilution and just plating. We ended up plating about 1/3 of the number of cells that could, so 40,000 divided by 3. And when the cells actually sit down and seed on the array, we got occupancy of about 6,000 colonies per array, which represents about 15% occupancy. So we definitely do have instances of doublets where more than one cell would settle in an individual well, and we'll deal with that later. But this, we optimized this as the best way that we could increase throughput and minimize doublet formation on the array. And so shown here is a bright field image where the G3BP1 signal is artificially colored in white to give you an example of what these cells look like when they're actually growing on the arrays. So the PDMS itself is not, cells can't grow on it. It's not conducive to cellular growth, but the individual microrafts are just like a cell culture dish. So when a cell sits down on there, it'll attach to the microraft and it'll grow and expand and form a nice colony on the array. And now if you let them overgrow, the cells will grow into neighboring wells and it'll go crazy, but this is about after 3 or 4 days of culture in lab. And so the first thing that we did when we adapted this platform was we had to make it compatible with high throughput imaging. So nobody had previously used these arrays with confocal imaging. They were actually developed as a method to do single cell isolation for single cell genomics. And so we had to kind of make some modifications to make it compatible with imaging. And the big modification and the big problem that we had to start out with is these arrays have a soft, flexible bottom. And that'll be important for something that I'll explain in more detail later. However, the huge problem with that is when you add media, it causes this major sag. As you can imagine, when you travel over the course of 1 square inch, this sag in the middle can cause major problems with out of focus issues on the microscope. And so here, we're just looking at some raft autofluorescence by blowing out the imaging parameters, and you can see this very characteristic sag when you look at the Y plane across the entire array. And so the way that we fixed this issue is we added a glass back behind the microRaft array and used that for imaging, and then removed the glass back later prior to selection of the individual colonies. So this was really helpful, to be able to flatten out this area and make this compatible with confocal imaging. So as we do our high throughput imaging, we're still going to use autofocus to help maintain in plane as much as possible, but at least now we have a flat surface that we're working with rather than the sag in the middle. And so to start this work, we used a positive control, which are guide RNAs targeting EIF2AK1, which is the kinase that is known to initiate the assembly of stress granules. And so what we can do is we can take cells that are labeled in 2 different colors. One has a non-targeting guide RNA and one targeting EIF2AK1. So in this setup, the green cells or the turquoise cells here would not form stress granules, and all the red cells will. And we can use that to kind of benchmark our imaging platforms on the arrays and see how it works. So when you take bulk cells and you use standard methods to quantify granules, you can see that either with regular 293 T cells or non-targeting guide, you have very robust formation of stress granules. And when you deplete EIF2AK1, you dramatically reduce the number of granules that are formed in cells. And this is comparable to a non-stress control. So these cells really don't form any granules at all. And so we use these cells combined on the array. And what you can see here from this zoomed in image of just 4 microrafts is that when you find cells that were expressing the red color, the non-targeting guide RNA, and you zoom in there, you can see that there are these distinct puncta that form in the cytoplasm of the cells. And that is in stark contrast to any of the colonies that we observed with the M-citrine color, which you can see over here, maintain that diffuse cytoplasmic localization. So we played around with the imaging parameters a bit, and we ultimately landed on doing our imaging at 10x magnification, and we used frame averaging to be able to increase our resolution. And then we kind of played around with increasing the resolution, but also maintaining the speed of imaging to go across the entire array. And so we can take these images and we quantify them from our pilot experiment we can see that all of the M. cherry colonies, or the vast majority of them, have a higher stress granule area relative to the M. citrine colonies, which have very low area overall. And we did identify with mixed color experiments or mixed color colonies doublets, and we can see very cleanly that those exist within the middle of these two different areas. However, you might notice that there is a lot of variability in the stress granule formation that we get across all these colonies. And so we're going to use some image classification later on to be able to deal with this issue and try to improve our calling of stress granules. So here's the total workflow of what we identified, and we call this process the Craft ID platform. So we're using a CRISPR-based method followed by microRaft cell culture and guide RNA identification to be able to identify guide RNAs that are contributing or that are targeting genes regulating a particular imaging-based phenotype. So again, the workflow in general is to infect cells in bulk, as previous methods have done. We decided to culture the cells for 7 days in bulk to allow those guides targeting lethal genes to be depleted from the pool. So we weren't interested, we didn't want to plate guide RNAs on the microrafts, which would then cause the colony to die, because we were only interested in screening ones that would be survived, or that would survive. I think if you were to take this method and design a custom library for this method, I wouldn't include positive controls of cell death because we don't want to analyze colonies that are not there. But given that this library has many applications for other uses, we decided to just culture in bulk for 7 days to allow for depletion of those lethal guides. We can then take these microRaft arrays, treat them with sodium arsenite for 1 hour, a very fixed and well-defined time point. And then you can, we performed fixation with formaldehyde and antibody staining, just as you would on a tissue slice or cell culture dish, followed by automated confocal imaging to be able to analyze localization of G3BP1.
Emily?
Yeah.
Can you say a brief word about the optical properties of the microRaft array, and then adding another 1 millimeter of glass on top of it, about what kind of objective you had to use, or if it mattered at all because you were using a rather low magnification anyway? Did this have any implications?
Yeah, so we played with a bunch of different microscopes, and having the 1 millimeter glass slide worked out at, we did 10x magnification. We could also have done 20x magnification had we wanted to, but we decided it wasn't useful to increase the amount of time it would take to go stepwise across the array by imaging in at a higher magnification. There may have been some microscopes that this was not compatible with. I don't remember everything that we had tried, but we used 2 different confocal microscopes, and it was compatible with both of those at the 1 millimeter. It was definitely a concern, and we wanted to use as thin of a piece of glass as possible to be able to get through it, and that worked out well.
Thank you.
So once we had this automated confocal imaging, the first thing that we had to do was be able to process all of these images. So all of the code and the software that we wrote to be able to handle this image processing is available on our GitHub repository. So we performed confocal imaging, and to get really high resolution of the granules, we imaged in 5 Z stacks. So we captured 5 different Z planes. We used 3 color channels. So if you have bright field, to be able to capture the grid, we had G3BP1, which was stained in red, and a nuclei marked in blue. And when we did this at 10x, it would cover 324 different individual stepwise positions, would cover the entire array. And this took about 6 hours of imaging time per array. So the way that this code works is we use this bright field image to be able to find individual squares based on this really well-defined pattern. And then we cut these images up and segmented them on the individual squares. We then filtered out ones based on the G3BP1 and nuclei staining to get rid of wells that were empty, because we're not interested in analyzing those. And so in total, we had over 5,000 unstressed colonies as a control, and over 119,000 distinct stressed colonies that we were analyzing. So all of these images we then took and filtered through a neural network to get rid of imaging-based artifacts, which I'll talk about in a little bit more detail on the next slide. And then we were left with colonies that we were interested in analyzing for area of puncta formation in the cytoplasm. So on this step, we used the very well-defined cell profiler functions to be able to accurately call and quantify the area of granules in the cytoplasm relative to total nuclei area as a control for colony size. And so we calculate this ratio of stress granule area over nuclei area and use that ratio to try to find ones that have a lower stress granule area overall. And so in total, we performed imaging across 20 arrays for our screen, which is a total of about 120,000 cell colonies. So if you remember, each target in our library has 10 guide RNAs targeting it. And we covered our whole library at about 10x coverage because there are 12,000 individual guide RNAs in our library. So in total, per given RBP, you can say that we have about 100x coverage at the guide RNA level relative to the number of colonies that we're imaging. So I think that with a lot of these imaging-based screening methods, coverage is much harder than using other platforms that are more compatible with high throughput, like simple growth rate or FACS-based screening. So it's definitely a delicate balance between the number of guide RNAs that you choose to use and the number of colonies or cells that you're willing to image and screen. And so the first thing that we noticed when we were filtering through all of these images is that they weren't all perfect, and there's a lot of imaging-based artifacts that occur when growing cells. I think this is true when you culture cells in a dish normally, or in our case, particularly on these microrafts, you have really small cells that are causing various artifacts. And so another graduate student in the lab, Noor, helped us to develop an image-based classifier using neural networks to try to remove all of these artifacts from our imaging, because as you can imagine, like in this example shown here of a lysed colony, if you try to quantify stress granule area on this lysed colony, it's going to say that there are no puncta, and in fact there are not, but that's not actually a phenotype that we're interested in using. So what we did is we manually curated images from the nuclei staining and categorized them into each of these different categories, which we found were most common. So sometimes we would have colonies that grew too fast, and so they're now out of the plane of focus, so our stress granules are blurry. There would be random debris. There were genetic knockouts where after treating with sodium arsenite, it would cause the cell colony to die. So we have this very characteristic cell lysis feature or phenotype that you can see from the very grainy nuclei staining. Sometimes we would get random rafts that are out of focus or having autofluorescence issues. All of that compared to this nice healthy colony where you can see really good resolution and separation of the nuclei in the colony. And so we used a one versus all approach where we manually curated images in these different categories, and we trained a classifier, and then we threw all of our images through each of these classifiers and see if we could assign them with high confidence to any one of these given categories. And if the image was assigned with high confidence to any given category, we classified it as such and then removed it from our analysis. And this was actually quite useful to help us filter out and find colonies with low stress granule area, because we removed over 17,000 images from this analysis. So this was about 15% of the data that we identified as falling into any number of these categories. And this is a little bit higher than the unstressed control. So the unstressed control was not treated with sodium arsenite, and we removed a fewer fraction of those overall. However, with the sodium arsenite treatment on top, that also induced a lot more of the cell lysis phenotype. So we had more colonies falling out of the analysis on that end. And if you're interested in seeing anything more about what went into the code to do this work, it's also available on our GitHub page. And so after we did this, removing all of the unwanted colonies, we looked at the stress granule to nuclei area of all stressed colonies compared to the unstressed colonies. And we identified a total of 101 that we deemed to be very healthy and looking good and have a low stress granule area overall. So you can see some images here. These are what our stress colonies would look like, and they have lots of granules forming in the cytoplasm with the quantification showed underneath. And these are some of the unstressed controls, so where we have a very diffuse localization pattern in the cytoplasm, and again, the quantification underneath. And shown here are 6 representative images of those 100 total, where we identified a reduced granule formation relative to the rest of the population. So you can see here that there's definitely some variability, and they aren't all entirely expressing no granules at all. Some are stronger than others. Some might even have doublets, like you can see here in this colony. A couple cells here have some granules and the rest do not. But we called these as overall reduced and lowered relative to the whole population.
Emily.
Yeah.
Yeah, so you ended up selecting 101 colonies out of the over 100,000 that you screened. So are these are really the only colonies that showed the phenotype that you were looking for, which is decreased cell granule area, basically, right?
Yeah. So we were originally kind of surprised, like expecting more RNA binding proteins to cause this like really dramatic no granule phenotype. But I think that we learned from this that knockout of a single RNA binding protein does not often have dramatic reduction in granule area. Our platform, we have a limited ability to call reduced area. There's kind of a what exactly is the cutoff of reduced by what percentage, et cetera. And I think given the variability in the quality of the image colonies across the whole arrays, as well as a lot of variability in granule formation, made it difficult to call a very strict, like reduced by 20%, et cetera, or something like that. So we just did the best we could to grab the lowest colonies overall that still seemed reasonably healthy. And then we decided to focus on that as kind of a positive selection. So if we take the lowest overall, what are the guide RNAs in there? And then what can we learn from that and validate in an independent manner?
Okay, so I have 2 questions following up on that. The first one being your quantification scheme would allow you to also identify cells that would have a higher granule to nucleus ratio. Is that correct?
Yeah, it would.
Did you look for those? Did you see any?
Yeah, we looked at those a little bit. We were less interested in those originally, so we didn't really focus on them. But the higher granule to nuclei area that we often found, the colonies also had this very distinctive kind of rounded formation. So it was like the colony itself was kind of rounding up and shrinking, and then the granules were really big in the cytoplasm there. And so we couldn't really figure out what that meant in terms of stress granule biology. We didn't find it particularly interesting because we thought it was more of an artifact of the total cellular behavior and not specifically the granule formation, because they did have that characteristic rounding phenotype as opposed to these flat dispersed colonies that we can see with the reduced granule formation.
Okay, and did you distinguish between the cells that had, or colonies that had lower granule to nucleus ratio based on the size of the granules or their number or the density of granules?
Yeah, again, that would be another phenotype that we're interested in looking at, but I don't know that the quantification on this platform wasn't high enough to be able to do that. So
It gets tricky
we
at this
did
point.
often see, we tried to look at the number of granules, and then again, depending on the particular colonies. So we imaged at a little bit lower resolution to be able to image faster. And so at that resolution that we chose, we could see diffuse versus puncta or not. But I think in order to actually quantify the total number of granules, we needed to go at a little bit higher resolution, which we didn't do for the screen. But that is a really interesting question because there's a lot of studies in figuring out how these granules may be fused together, or what's essentially kind of actually nucleating stress granules versus allowing them to continue to grow and get bigger in the cytoplasm. So those are really interesting questions that we're interested in addressing, but we didn't capture them at the imaging resolution that we chose to use here.
Right,
yeah, if I were to do that again, I would go with a higher resolution and then maybe fewer guide RNAs in the screen to be able to maintain the coverage.
So would you be able to quantify on the fly, so as you're imaging a given raft, and then say, if you pass a certain cutoff, then you go higher resolution?
We didn't try that. That's an interesting idea. So the way that we did it is we captured a whole raft, and then we took that data actually just to a separate computer and then
Right.
processed it, because we were kind of imaging and developing the pipeline at the same time. But presumably you could, if you're on the same computer and as an image is acquired, pipe it into your analysis and then basically say whether or not you needed to go in. That would involve way more manipulation of the imaging software than I'm
Right.
familiar with. But yeah, in theory, I think that seems totally feasible. That would be really cool.
Okay.
Okay, so I want to take a minute to kind of highlight why we chose to use these rafts in the first place and what I think is kind of one of the coolest features of them, and that is the ability to physically isolate the colony when you have something selected of interest. So we use that glass back for imaging purposes, and then you can remove it from the back of the array once you're done with your imaging, because what we wanted to do was physically isolate rafts that we were interested in for PCR-based sequencing analysis. So the array is soft and flexible so that a microneedle that you put over a microscope objective can be used to poke an individual raft out of the array. And these rafts have magnetic particles in them, so once you poke it out, you can use a magnetic wand to pick it up and then move it over into a PCR dish. So this setup and system is quite simple. I think that the needle that goes over the objective, you can buy it from Cell Microsystems for, I believe it's $1,200, at least that's what it was like 4 or 5 years ago. And this process takes about, I would say, 1 to 2 minutes of actually moving the array to the position that you're interested in, poking it out with the needle and picking it up with the micro wand, about 1 to 2 minutes to collect an individual raft in a PCR tube. So this again is a limitation in terms of throughput, where we were able to select 100 colonies, over the course of like 2 days or so. But as you have to spend about 2 minutes collecting each one, I think that collecting, thousands of colonies would maybe be difficult with this method. However, the Albritton lab and the engineering labs who developed these arrays, they now have robotics platforms to be able to do this robotically, but of course that's a little bit more expensive and requires specific machinery. So what we then did is we took these rafts that were isolated into DNA lysis buffer, and we performed a 2-step PCR using barcoded primers to be able to use next generation sequencing and then deconvolute what the guide RNA was infected in each colony, where we have a unique barcoding system to be able to match that back up to the original colony that we collected. And so keep in mind that we're isolating DNA from a very small colony of cells. We've only got about 5 to 20 cells on a given microRaft array. And the other issue is that these cells are fixed in PFA. So we are getting DNA out of fixed cells. And so this was a little bit challenging to get the sequencing to be able to work with this PCR-based approach. So in total of the 101 that we sequenced, or that we picked, only 61 successfully sequenced with this approach. And then we were able to validate 38 of those independently using siRNA-based depletion. And so since this work has been done, we've made some improvements to the library prep method, and further optimization can be done to kind of boost this 61 closer to 101 using more efficient DNA lysis and PCR-based methods. But what we did show is that we then took the ones that we sequenced, the guide RNAs that we identified, and we independently use siRNA-based depletion as a different method to be able to validate that these RNA binding proteins do in fact reduce stress granule area. So shown here along the x-axis are all of the different targets that we've identified relative to a non-targeting control all the way over here on the right, and the unstressed control on the left. And then plotted on the y-axis is the normalized stress granule area normalized to the non-targeting control at 1. So keep in mind we're using siRNAs here, which will deplete the total protein, but it's not a genetic knockout like a CRISPR-based approach is. But this allowed us to check a bunch of these things very quickly. And what you can see is our positive control, EIF28K1, as well as G3BP1 are the most depleted in this method. Shown in stars down here are ones that have previously been characterized to reduce granule area formation overall. And then listed along here are a bunch of new ones that we identified do significantly reduce granule formation and reproducibly across multiple replicates. And so something that was really interesting, we found a lot of splicing factors in this work, which was a little bit unexpected, given that these stress granule assemblies are often associated with translational components and not just splicing, or not splicing components. So that was pretty cool. We also identified a lot of ribosomal subunits as expected, given that stress granules, again, have these translation assemblies that are being halted in the cytoplasm, as well as some mitochondrial associated subunits. And you can see that there's definitely a degree of variability in the total amount of stress granules that can be reduced by depletion of a single RNA binding protein. And again, this was pretty surprising, that we didn't find anything else that would deplete it as strongly as this kinase, although I guess maybe not too surprising given that that's essential for initiating this cascade. But this goes to show that manipulation of a single RNA binding protein in isolation is not enough to totally deplete granules in the cells. But shown here are what the siRNA validations looked like for 3 of our strongest novel candidates. So these are 3 splicing factors and a zinc finger protein, which when we performed siRNA depletion on a plate, we saw a reduction by about 40 to 50% of the total granule area relative to a non-targeting control. And I thought these were pretty interesting because there have been papers associating these proteins that have genetic variation that are associated with various neurological diseases. So SNRMP200, for instance, has a point mutation that is often found in the genome of patients with retinitis pigmentosa. PUF60 has genetic variations, again, in the genome that are associated with microcephaly phenotypes. So again, a different neurological disorder. So it is possible that maybe regulation of size and formation of granules under those genetic conditions is related to what those diseases might be doing. But of course, that takes a lot more validation and work to be able to follow up on. And this has also led us to some interesting studies or thoughts about how manipulation of splicing factor proteins maybe is affecting the transcriptome or the alternative splicing landscape, which is then essential for regulation of these stress granule assemblies, but we haven't done any of those further validation experiments. And so in total, I'd like to highlight a summary of what all of the different proteins were that we found to deplete stress granule area. And what you can see with this protein-protein interaction plot is that when you look at their association rates with either UBEP2L or G3BP1, which are kind of core components of stress granules, many of the components that we identified are found via mass spec or studies to have protein-protein interactions in the granules. So many things that we found definitely exist within the granules themselves. However, we also identified proteins that exist outside of granules, and these would represent either upstream or downstream regulators of granule assemblies that some of our mass spec, if we were to only focus on hits from mass spec, we would have missed in being able to assign the regulation-based phenotype. So I'd like to highlight a few takeaways from kind of what we learned from doing this work, and if I were to apply this screen in a future method, some sort of considerations that I would take before deciding if this was like the best platform or not. So something that's really nice about using these microRaft arrays is that they are simple to easy to use. If you can culture cells on a dish, you can culture them on microRaft arrays, no problem. The other huge benefit of these arrays is that the cell colony can be isolated from the array. This is a unique feature of this platform that you can't get when you just do in situ imaging on a dish. And so we chose to do imaging on fixed cells because stress granule formation is transient and we had to stop it after a certain time point. But if you had a live-based imaging reporter or a live-based phenotype, you could isolate the live colony out of the cells and continue propagating it for various downstream assays. So that I think is a really strong feature of this array. It doesn't have to be a fixed time point. You can isolate your colony and continue culturing it. The other nice thing is you can use standard confocal microscopy. We've imaged these arrays on a number of different focal microscopes at this point. The setup is pretty simple and the image processing is pretty simple because the grid pattern is so well defined and makes it easy to segment these images. Given the limitations in isolating individual microrafts from the array, I think that this works best for something like a rare phenotype where you're not hoping to isolate tens of thousands, but rather just a couple hundred, which makes it a little bit easier to recover the cells that you're interested in. And then considerations of cells that will grow well on small surface areas. So you can't do this on really large cells, or you could, but you would want to change the size of the microrafts that you're using. And then again, imaging-based considerations. It's limited in throughput compared to in situ sequencing, where in our method you have to specifically isolate a given colony to sequence it, as opposed to sequencing all of them at once. However, we don't have to do in situ sequencing and imaging on a plate, so that makes our lives a little bit easier. And again, with the scaling issues, I think that this works best with targeted libraries, and it's not necessarily scalable to the genome size. So before I open up for any more questions, I just want to thank all the people who are involved in this work. This is the YoLab, and many people have played a huge role in this project. As you can see, we're quite a large group, and we work together a lot, and I had fantastic input from many lab members along the way. To my mentor, Jean Yo, who supported and helped with all this work. My co-leader of this project, Anthony Vu. This is really something that was led jointly between the two of us and would not have been able to be accomplished with either one of us in isolation. Jackie in the YoLab, she developed the CRISPR guide RNA library that we used for the screen. Nora helped a lot with the image analysis classification, and the Albritton lab helped us with the glass back modifications and helping teach us how to image on these microRaft arrays. Her graduate student, Matt DeSalvo, was really helpful and patient in this work. So thanks so much. I'll just go back to here, and if there's any further questions, I'd be happy to take them.
Yeah, thank you very much, Emily. That was great. Very interesting platform. So you just mentioned here in the takeaways that you think that the platform is best suited for rare phenotypes. Is that just because of the work that's involved in picking up a single raft and then extracting and preparing library out of these rafts? And why do you think that is?
Yeah, so extracting and then preparing libraries for each one is time consuming. So you can use, I think that if there were less of a rare phenotype that you're interested in, using some robotics platforms to help isolate these things with a machine rather than doing it individually would be better. Because as I mentioned, isolation of each microRaft takes about 1 minute to 2 minutes, so that doesn't scale very quickly. And if you have something that's a stronger phenotype and representing a bigger fraction of your population, it might be more beneficial to do in situ sequencing, where you capture the guide RNAs for those alongside of the imaging. But again, then you have to do in situ imaging.
Okay, so to be clear, in this setup, you were actually picking the single rafts by hand. You were not helped by any robot.
Correct. Yeah. So we did it by hand, and in the case if you have fixed arrays, it's pretty easy to just you put them in the fridge and keep picking whenever you want. You don't have to do it all at once. So we were able to pick like 100 in the course of, I think, maybe 2 days. But if you were to pick over more time frame from that, or if you get yourself a little undergrad army, then you can pick as many as you want.
So talking about the fixation step, I mean, presumably that would affect the efficiency of your DNA extraction and how feasible it is to prepare libraries from that RNA. So what fixative was it just formaldehyde?
Yeah, so we used 4% PFA fixation. And yes, we do believe that it had an effect on the efficiency of DNA extraction. And also the fact that, we're amplifying from only 1 copy of lentivirus per cell,
right.
And we've got like 5 cells, 10 cells. So the starting material is very, very low. So what we ended up doing was using, a PCR-free hood to prepare these libraries so that we could use high numbers of amplification. So I think we did, in our 2-step approach, we used 22 cycles of amplification for the first round and like 10 for the second round. And so by doing that in a clean hood, we could do that very well without having contaminants. But when you do that in a open space, there were a lot of contaminants that came into play there. So there's definitely some optimization that can be done to improve that step.
Okay, now more of a biological question. To me, it was a bit of a surprise that you only found 101 colonies out of the 100,000 that you screened that gave you a phenotype, and we touched on that earlier. So do do you think that it's only about interactions between different RNA binding proteins regulating this process of stress granule formation? Or is it just that there are very few players involved in the formation of these granules? And also to test this hypothesis that you have multiple RNA binding proteins involved in the formation of the granules, how about using pooled guide RNAs or a higher multiplicity of infections so that you have a number of guide RNAs transducing each colony?
Yeah, so I think that to your second point, it would be if we wanted to test these multiplicity things, what I would recommend doing is using the CRISPR vectors that allow you to specifically design and express more than one guide RNA on the vector itself, so that you can very nicely control for that and design it in a way that you can recover rationally combinatorial design. In terms of if a single RNA binding protein can strongly reduce these granules, the fact that the granules themselves have like hundreds of proteins inside of them, I guess it's not that surprising that depletion of a single one would really ablate them completely. And since that's kind of what we were looking for in our screen, is the most dramatic reduction phenotypes, it makes a lot of sense that like our positive control and G3BP1 itself were the strongest regulators of depletion. And we and others have shown that, for instance, that UBEP2L will deplete them again at like a level of 50% in a various different degrees of depletion overall. So I don't think it's that surprising given how many protein components there are in these stress granules. And I think that it's also cell type and context dependent. So to be able to figure out, you know, in more detail, whether or not any of these proteins would have an effect in vivo, I think it'd be really important to look specifically at how these granules are forming in an in vivo setting in the context of maybe a more natural stressor besides sodium arsenite, to see whether or not the effect of depletion of these proteins would be enough to prevent this overaggregation phenotype.
Yeah, what about the possible implications of non-RNA binding proteins or proteins that are not necessarily known for their RNA binding properties? Maybe, you know, proteins with highly disordered domains that tend to form self-aggregates or membrane-less organelles.
Yeah, I mean, it's definitely been known that these granules are enriched for proteins with IDRs. And so I think that designing a targeted library to proteins with IDRs beyond just RNA binding proteins would also be really cool.
Yes.
Yeah.
Cool, very interesting. I think that we'll stop here. We're just past the